Welcome to the Epigenetics Podcast from Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Lothar Schermelleh from the University of Oxford on this show. Please let me briefly introduce you to our audience. You got your PhD from the Ludwig-Maximilians-Universität in Munich in 2003 and stayed there for your postdoc. You then moved to the University of Oxford in 2011 as a senior research fellow and became associate professor there in 2020. And you are still there today. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and in pursuing a career in science?
Well, I was always interested in natural sciences, although not particular biology. I really enjoyed at school geography and physics and all sorts of things. And so I knew I wanted, I had a desire to go into science, although I have to say I'm completely from a background without any kind of I'm a first-time student in my family, so no
first-time academics, so to say.
Yeah, so no. So basically, I haven't had any role model or anything to follow. So basically, I stumbled into studying biology more or less by accident. Architecture was another interest of mine, so that didn't turn out. So I started biology, and I would say of interest, but without any idea what I'm going to do, as a profession or anything. So that only developed far later, when I sort of got a glimpse into what actually the life of a scientist could be.
What it meant to do an academic career, right?
Yeah, exactly. So that was an accident.
Yeah, that's I hear that quite often, that it was not planned, and I just went with the flow and what interests me, and then I'm here where I am right now. So
yeah.
So
it turns out. Oh, please.
Yeah, go ahead.
No, it's just also kind of reminiscing about the time. I was never brilliant student or so. I mean, I got around quite all right, but never in the top tier. And it was only when I first time really joined a lab for a project and got exposed to more than just the theory of science. When I realized, okay, that's something I can relate to and maybe contribute to, and that's basically when I really started to develop a passion. I have to say, before it was a bit more kind of tedious, but that really opened my eyes. So that this is something I would like to continue doing.
Yeah, I had the same feeling that the more I got into like the real practice of doing science and the real projects and the real goals that you're going through, then it got more interesting and really more exciting in the end. But let's come to your science that centers around understanding rules and dynamic regulation of three-dimensional chromatin organization governing mammalian genome function using advanced super-resolution optical imaging solutions. I guess this, I prepared this like several weeks ago, but I guess I took this from your website. So probably. I want to start with your
was never meant to be read out. No.
I want to start with your... sorry.
No, please.
I want to start with your first major publication, which was published in Science in 2008. There you applied three-dimensional structured illumination microscopy, which is 3D SIM, or short 3D SIM, to circumvent the intrinsic limit of spatial resolution of microscopy to study the mammalian nucleus. There you imaged chromatin, nuclear lamina, and the nuclear pore complex. Can you maybe give us a short summary to higher resolution microscopy, and then talk about what you were able to find in the study?
Mm-hmm. Well, generally, optical microscopy, so as opposed to electron microscopy, is fundamentally limited by the diffraction laws of diffraction, so the diffraction limit. It basically restricts the ability to resolve details to about 200 nanometer at best. And of course, most molecules or molecular complexes are smaller than that. So there has always been, electron microscopy as a means to overcome the limits of light microscopy. But that always involved, of course, fixation of the samples and very harsh treatments. And in the end, you got a gray scale picture of something without really knowing what you're looking at. So that was good for certain aspects, membranes, maybe viruses, but most of the actual interesting biological structures and complexes were really hard to address that. So basically, it was always kind of a dream to overcome this limit, to bridge the gap to electron microscopy and profit from all the benefits of light microscopy, specifically fluorescence microscopy, so you can really specifically label individual molecules and complexes. To overcome the limit of resolution. And by the time I started to delve into that area, there have just been the first techniques being developed that enabled to overcome this limit. And one of these techniques was structured illumination microscopy. And I was just lucky enough to meet one of the developers of this technique right when they were, rolling out a prototype. And there was John Sedat in San Francisco. And he, together with Mats Gustafsson, have developed this technique. And I basically, that was at the time a complete side project. I met John in Munich. He gave a talk there. We discussed, we had some kind of interest in trying this out because I was interested in microscopy for a long time. And basically, we applied for a little travel grant, 5,000 euros at that time. So me and my supervisor, Heinrich Leonhardt. And I traveled there with my samples and happened to be the first that put biological samples under this microscope. I worked, basically as a visiting scientist with the developers together and tried to get the best out of my samples. And they turned out to be just ideal to showcase the power of this technology to be able to resolve, individual nuclear pores where they sit in the nuclear kind of envelope, how chromatin organizes around those nuclear pores, how they basically pinch a hole into chromatin and give access to chromatin or channels within chromatin. And, we basically, it was like lifting a veil. Before we were essentially just looking at blur in a way. And it was based on, our fantasy, what we make of it. And suddenly things become completely clear and obvious and also surprising in a way with this. So in this first application, basically, we showcased what this technology can do, and that was basically turned out to be a science paper, which was the best 5,000 euros ever spent.
Definitely. So what is it that makes it so special, this technique, in comparison to those methods that were around, and what was the major advancement?
Well, first of all, at that time, principles were there to achieve even higher resolution. There was localization microscopy came out around the same time. Storm, palm type of imaging. And a few years before, there was already STET being developed by Stefan Hell, and that all kind of showed already kind of on ideal samples what can be done. But with the technique that we've used, we were able to do this right away in 3 dimensions. So to do 3-dimensional imaging with 3 colors, with, we ticked a lot of boxes that at that point haven't been addressed. So that sort of, came a bit as a surprise, I think, in the field that we could achieve this, and already make, valid or proper biological discoveries and quantifications of aspects of nuclear organization that wasn't possible before.
So did you only image proteins, or was it also DNA, or did you just do it all?
Well, I was always interested in chromosomal organization. My PhD was actually in the lab of Thomas Kremer, who is one of the pioneers in studying the organization of chromosomes, using fluorescence in situ hybridization, these kinds of techniques. But that came from a cytogenetics kind of side of things. And during the time when I started using super resolution imaging, I was pretty much more focused on epigenetic mechanisms, on how enzymes, specifically DNA methylation enzymes, work and are regulated. So I basically came back, through the back door on, this, my previous passion for chromatin. So what I want to say is, yes, we did image chromatin just by looking at, the standard DAPI counter stain, in addition with immunostaining of the lamina using lamin B antibodies and nuclear pore complex, which was not, it wasn't with a particular kind of aim. It just seemed like an appropriate starting point for us, because we knew how to do these labelings really good, and preserve the cellular morphology in the best possible way.
What you then did was looking at the inactive X and how bar bodies are formed at the onset of inactivation in early differentiating mouse embryonic stem cells. So was that then the next logical step to find a functional thing, looking at heterochromatin? And what did you find there?
Yeah, absolutely. I mean, the X chromosome, or it's a classical model for epigenetic gene silencing, and at this point, I mean, we were basically elaborating more on how can we better map active and inactive areas of the genome within this nuclear landscape. And the inactive X chromosome was one of the kind of targets. What we discovered essentially was that when we localized the cis RNA, which is like the main regulator of this X inactivation process, is this has always been described by that time as a cloud. People were discussing whether it sort of surrounds the the inactive X chromosome, or what. But basically, by optical imaging, it just looks like a cloud essentially, and we could, of course, with our increased resolution and this increased three-dimensional sectioning capability, really pinpoint individual foci of cyst RNA, and we could count them, and we could locate them in particular pockets within the otherwise compacted X chromosome. So we could basically, again, in a way, generate a new and refined image of something that has been observed for a long time, but not really interpreted in the right way. So that basically then created the fundament for, looking into different aspects or thinking about this process in a different way. And we followed up with, of course, our collaborator in Oxford, Neil Brockdorff, who's, of course, one of the pioneers in that research, to look at the mechanisms, how these molecules, find the right targets and spread out during the onset of X inactivation, but sort of keep to the X chromosome and not kind of continue working, all these kinds of things. So basically we started really to use our advanced imaging to use more and more in a quantitative manner, but also really as a readout tool for functional assays. So that's basically where we are now on that end.
So for that, you also needed to do RNA fish, I guess, then, right?
That is true. Yeah, RNA fish is classic.
So is the resolution of those DNA, RNA, so all those nucleic acid binding fluorescent images or assays, is there another layer of resolution based on, because you're using DNA? So is it advantageous to go if you improve the resolution on the microscopy side, or is it intrinsically to the assay that you maybe don't get a better resolution there?
That is an excellent question, because people always think about, how much resolution is enough, or they rather say, oh, I need more resolution, even though they might not even need it. So you always have to, of course, keep things in, at the right, think along the right scales, and then find solutions that will give you an answer to your biological question. So meanwhile, we of course combine other techniques as well. We do single molecule localization when it's more appropriate. But we do still do a lot with structured illumination. Microscopy, because it turns out for many biological questions just about the right level that we need, and has a lot of advantages when it comes to multicolor 3D live cell imaging, and so we profit from that. So you've asked about hybridization techniques, RNA fish, DNA fish. That is a very crucial technique when we, of course, look at the organization of specific sequences within the genome. The problem has been, or still is to some extent, that the technique, to enable to bind specific DNA probes, which is like requires a denaturation of the DNA, typically done by heating and under kind of harsh conditions that will ultimately destroy the ultrastructure that you're interested in. So far, it has been acceptable because, of course, the resolution was not there. So when people used fish techniques, they were interested in sort of, distance relationships on larger scale. But of course, if we want to look at the underlying chromatin organization, we don't want to, cook this beforehand. So we lose a lot of the subtleties of the organization. So that's why I was also involved in improving this technology. And again, I don't want to claim any credits for this, but colleagues in Oxford, Veronica Buckle and Jill Brown, have developed this technique called razorfish that basically avoids this kind of heating denaturation step and uses an enzymatic digestion of DNA to like an exonuclease digestion to create single-stranded DNA that then offers this target to probes, and that doesn't destroy our ultrastructure and allows us to do this in a much more gentle way, and then again profit from that better resolution. Right? Yeah. Before, we were just destroying things, and then that didn't matter. Really, now we can basically put this together and really profit from the resolution increase. So, but basically what I want to say is like the development of labeling techniques go hand in hand with the kind of optics development side. And otherwise, I think most of the leaps in recent time really came from development of probes and labeling techniques rather than the optics side.
Yeah, I want to come to another labeling technique that you are involved with just in a minute. But before we go to that, I have another question, because when I started doing back in the days fluorescence microscopy, I was looking for objective control parameters, right? What can you do to control your experiment? Am I exposing long enough or short enough, whatever it might be? And there you published SimCheck, which is an image shape plugin, which makes it possible to QC your high resolution images, right? So how did you get into that? And what are those parameters that you use as control parameters, basically?
Well, first of all, how did I get? I'm a stickler for. I can really. I'm not content with just an image that shows about what I want to see. I want to have it perfect, and I quickly found out that this technique that we are using, structured illumination microscopy, which requires some complex mathematical reconstruction parameters, processing, is sensitive to artifacts. It's prone to artifact if certain aspects in this imaging process go wrong. But of course, at that point, in order to really identify those, you needed a lot of experience and a good trained eye. And I realized what many colleagues just didn't understand or could differentiate between artifacts and kind of real biological structures. That created a lot of confusions in the early days. And I realized, okay, with some knowledge of the imaging process and the principles behind, we can come up with some specific quality control metrics that can help people that apply this technique but don't necessarily know the background, still measure the quality of their data and then use this to counteract potential problems. So specifically with SIM is, for instance, that you can only extract this small details, the information, which are basically high frequencies in an image that you basically can extract because you create an interference of your striped illumination with the light that is exposed or emitted from the sample. Anyway, this only works, this Moire effect, when you have good stripe modulation. If you see stripes in your image, then the more you see that, the better your math works. And so basically, we devised a method how we can map the strength of these stripes in the raw data and then decide the goodness of the reconstruction in different areas of the sample. For instance, there are many more. Also, kind of measuring resolution based on the frequency extent and so forth. So, that has been quite successful, and many people have been using this tool because, of course, companies don't have that in their, integrated in their software. They don't really care.
So what you did next is describe refined methods for pulse chase replication labeling of individual chromosome territories and replication domain units in mammalian cell nuclei with a specific focus on their application to a three-dimensional structure, 3D sim, basically. So what is important when labeling cells for those kinds of 3D experiments?
Okay. Well, these... you have to play with tricks if you want to, in this case, say you want to understand the dynamic process and progression of DNA replication and what it does to the local environment. Sometimes you cannot just trace the process in a live cell situation because it takes too long, too much, you have to pay a price for imaging repeated time points and so forth in 3D and so forth. So pulse experiments are a good way to extract dynamic information on fixed samples and still get the best possible imaging quality that you need for quantitative evaluations. So with that said, so essentially you can use different reagents or different precursors of DNA replication that are incorporated at different times that you add to the cells while they replicate. And
you basically create a memory, right? Of what's happening.
In a way. Yeah. And you can extend this in various ways. You can also use this technique to label, let's say, active chromatin that's actively transcribing chromatin that's replicated early in S phase from heterochromatin, and observe the fate of these two chromatin types during several cell cycles. If you like, you can use this technique to label individual chromosome territories and assess their dynamic properties and so forth. More recently, we've basically adopted this technique for RNA, exist RNA labeling. So we used, again, it's the development of labeling techniques that often give you that kind of new information, but there we used halo tagging of an RNA binding protein, for which we created a stem loop kind of tag to the exist RNA. And then Halo is a kind of a self-labeling little protein tag that you can add a ligand to it. So it will basically covalently bind a fluorescent ligand to it. And then once it's labeled, it's there. You can observe the fate, either in a live cell situation, but you can also just leave it there, kind of stop the labeling, and then label with a different color after a certain chase time, and then basically distinguish between kind of older molecules and those that are just being synthesized. And you can see how these two kind of cyst or RNA molecule types interact with each other, or you can infer some rules of how they use each other, how they bind over time, and extract dynamic information from actually fixed cell imaging. So that has been quite successful. But you see, I'm tinkering a lot around, how can we use certain labeling techniques to our advantage, but always with the goal, okay, how can we address this particular question? And
that is a good segue into my next question, because you tinkered even further in 2020, where you used live at fixed cell 3D super resolution and scanning electron microscopy in this case to analyze structural and functional nuclear organization in somatic cells, and identified chromatin domains at the 200 to 300 nanometer scale. The structure of chromatin at smaller scales, or at this scale, is still a matter of kind of a debate, right? I mean, it's still not clear and people have different opinions. So what did you find there?
That's interesting, because that size scale, I would say, anywhere between 100 and maybe a few hundred, 200, 300 nanometer is exactly that sort of blind spot of any imaging sort of approach. Of course, we know from electron microscopy since, many decades, the beads on a string conformation, of course, yeah. And even though that was in purified chromatin, the assumption is that a lot of chromatin within the nucleus is exactly in that sort of bead on a string conformation. So nucleosomes that are evenly spaced and form a filament, a fiber, where then, transcription occurs. But, fact of the matter is that these nucleosomes interact with each other and with other proteins to form higher ordered structures. And this is often underappreciated in kind of molecular biology, because basically it's just simple to assume an open chromatin that's bead on a string and a closed chromatin, heterochromatin, that's basically inactive. But things are more complicated, more nuanced, and that's what we already saw since quite some time. With our super resolution technique, we were able to observe distinct domains with relatively sharp edges and pretty, distinct areas between domains that is apparently free from DNA. So this is what we call interchromatin domains. Of course, other people, colleagues use different techniques. They see something else. So it's always like a matter of, what technique do you use? How do you, and I don't mean it in a disrespectful way, but how do you describe that sort of landscape? How, where all the action happens? Yeah, is that simply open chromatin and closed chromatin? Now, of course, we know since some time that chromatin organizes in loops. Cohesin is one of the factors involved in generating sort of loops that are kind of hold together on their basis by cohesin and CTCF. Never mind. So there's more to it. But anyway, we haven't had that sort of clear view to it. Now we were thinking, okay, with our structured illumination, because it's really it's inherently three-dimensional, and we can really image under very challenging conditions, because the whole nucleus is full with chromatin, and it's lots of fluorescence, and it's challenging from an imaging perspective. But of course, people will always doubt, because the SIM is not the highest resolving technique, and so we had to find a way to complement our findings. And at that point, I came across, the recent work from our colleagues from Genelia. Harold Hess was driving a fantastic development to establish 3D, electron microscopy using focused ion beam milling, and then surface electron microscopy on large, like entire cells, essentially. They did it on cryo-fixed samples. And this is really important, because usually when you do like the classic 3D EM approach was always chemical fixation and then dehydration, resin embedding, and sectioning. But there are always doubts about the effects of these relatively strong fixing reagents. So turns out that the cryofixation and the approach that Harold Hess and colleagues have developed was very, very in line with what we saw, like distinct domains, assemblies of nucleosomes. So this time, we basically use the same cells or data sets from the same cells that we were using, and you could observe assemblies of nucleosomes that form like distinct domain features that are then separated by this interchromatin space. So suddenly we had a way to correlate our findings with a technique that is inherently higher resolving, but basically showed the same thing. So at the moment, I'm quite confident that these kind of 200 nanodomains are really physical subunits of chromatin, and not only of heterochromatin, but also of euchromatic areas. Because what we also kind of see is that all the transcriptional activity occurs on the outside of these domains, whereas heterochromatic marks are further, are located in the interior of those domains. So there seems to be kind of a sorting out of active and inactive chromatin on a rather small scale that we didn't expect.
Yeah, that sounds very interesting. So another thing that has come up lately is artificial intelligence. And I've also seen that some microscopy approaches would use that in their approach. Are you also implementing this into your approaches?
We're observing the field, I would say, because of course, it's powerful, and it can be used for imaging, of course, in many ways, for the good and for the bad, I believe. So as always, when new tools come around, so I think it has a lot of potential. We haven't come around to implement those yet. But what one risk really is to use AI tools or deep learning to basically overcome, basically to lower the standards on your sample and your kind of classical kind of standard acquisition. So not to get further, but basically to lower your standard, make it more, less difficult for people to come to the same results. And then, it's there are the classical risk of course, the dependency on the training data, the risk of hallucinations, and how much you interpret into this result. So I think there's still a lot to learn. But yeah, it's always how you apply it, and not using it as a shortcut, but like as a next level.
So imagine you are due to submitting a grant proposal tomorrow, what you maybe are. What would you have written into that grant proposal?
Oh, brilliant. Yeah, I've just submitted one last week. That was actually for a microscope development or microscope technology development project. And this is to establish super resolution imaging under cryo conditions. So cryo fixed samples and implementing adaptive optics to it. So adaptive optics is a very powerful method to overcome, aberrations that are induced by the sample or by, various issues with the sample. It's a particular problem with cryo imaging under cryo conditions, because you basically look on vitrified eyes or cells embedded in vitrified eyes, which is basically like glass. So you have a little lens that you're looking onto, and that creates problems. So in this way, we want to enable like artifact-free super resolution imaging of cryofixed samples, so where we absolutely know that their structure is perfectly preserved, and then take those samples to cryo-EM imaging, so either cryo-EM tomography or cryo-FIB-SEM, and use the data from the super resolution data from the light microscopy to correlate it with the electron microscopy data, provide the necessary context, allow us to pinpoint, certain molecules or configurations of macromolecules. So that is one thing that we're interested in. I think there's a big push towards correlative imaging, and I think what we can contribute there is the implementation of adaptive optics, also the quality control aspect that we talked about, auto calibration, and making these things more user friendly, more reliable, and also of course implementing AI tools to get the best out of it. So.
Yeah, that sounds very demanding and very interesting, and also very expensive.
Yeah, it is in a way, but you always have to look at physics labs and whatnot. In comparison, we're still cheap science.
That's true. So for the last 45 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Let me think. No, I think you've covered a lot. I think where we really operate is on that interface of technology development and biological application. So I think this is really important, that you know why you develop certain things, because you want to address and answer biological questions. I think that's sort of the theme that I'm trying to follow.
Okay, so thank you, Lothar, for your time and for being on this show.
Thank you for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.